know, this is the first time we come to Egypt. We are very proud of the chairs, and we estimate to arrive in March. We know that there are many discomforts, many groundless, and also this morning there is much delay. We are sorry for this, and we hope that this will not occur next time. Also, I would like to welcome our dear friend, from German University of Cairo. She shared with us many scientific activities. He is chairman of biochemistry in pharmacy college of UC. He shares us in many activities and you are welcome too much here in Mansoura. We know that you are busy, you have many duties, you have many work in your college, and we estimate you all so much. Also, I would like to welcome our guest from African students who shared the activity, who came to us, to our brothers, all African countries who are proud to be together, and we hope to cooperate, and we hope to stay in Egypt and to gain some skills from sharing this activity. Also, I welcome the Egyptian students who are sharing this activity. Also, I would like to send my deepest thanks to our dear friends from universities of Delta, to the Catholic University, Professor Dr. Yusreya. She is busy in her section, however, she insisted to share with a good connection of her pupils and her colleagues. Also, I extend my deep thanks to Fanta University, Professor Behel and his colleagues, and hoping that we will enjoy a very good meeting today. Our estimated guest, Professor Brown, was from Queen's Square, London, to present us. I have no presentation. There is a theoretical lecture. Then we have a clinical meeting for some extrapyramidal and movement disorder piece. Professor Harris.
Can you give me the back?
Yes,
okay. So, it's a great pleasure to be here. It's very kind of Ibro and Human Society to invite me here. Very grateful to Dr. Well Mohammed Salama for organising this meeting. It's fantastic to see so many people from all over Africa here at this meeting today, and it's very kind to be welcome to the Neurology Department in Mansoura University today and have a tour of the facility and see what's happening. So it's very kind of all of you to put this meeting together and to invite me here. So I've got about an hour to talk. What I'm going to do to start with is just to talk generally about genetic approaches to neurological disease. So this has been since about 1992, when the first gene was identified for autosomal dominant Alzheimer's disease. This has been a driving force in neurological research. I'll talk a bit about the hypothesis behind that. One of the important issues is whether families of neurological disease can also tell us about people who don't have a family history. So this is a really key issue for all of us in the field who are interested in trying to develop new treatments for our patients. The other issue that I want to really try to illustrate in this talk, particularly being here with you in Egypt is this genetic study of disease is about people and it's about places. It's about different places around the world. I'm afraid to say that I think that to date, most of the research that has been done on neurological genetics has been based on patient populations in Western Europe and North America. And there is a huge amount to learn in other parts of the world which we've barely scratched the surface of. So I think working together, we have a huge amount to learn about these diseases in different parts of the world. So I am a Welshman. So within Great Britain, there is a separate country which is called Wales. This is separate to England. This is something particularly my American colleagues find difficult to comprehend. So I trained in London at Queen Square, and then for 10 years I worked in South Wales. So Wales is a separate country from England. It has its own language. It has its own sporting teams. It's a separate entity. But many of the families that I'm going to talk about are based in this area in Wales. A year ago I moved back to Queen Square to pursue my research further. But a lot of what I'm going to talk about is based around Wales. So for those of you familiar with the UK, this is the sticky out bit on the side. Down here is Cardiff, the capital where I was based. This is West Wales. These are areas of people like myself, who are the Celtic people on the fringe of the UK, who have a separate genetic background to the English Anglo-Saxon people in England. And I think this is important. The other message I'd like to give you, particularly the IBRO students who are perhaps on the start of their research career, is how important research into these diseases is. So this is a quote from Professor Bob Williamson, Melbourne University in Australia. So this is very much, I think after he'd been doing battle with the ethics committees who sometimes try to frustrate our research, to say that research is not a neutral activity. Research is a contract between the researcher, the community, the patient, their loved ones, to try to find an answer for their disease and to develop new treatments. So research is a very important part of what we do, both in terms of developing treatments for disease and understanding biology and how biological systems work. This is a very important thing that you're doing and you're embarking on. So in my talk I'm going to cover some of the general principles of neurogenetics, the types of studies we can do, which I think are very straightforward, but sometimes obscured with jargon and terminology. I'm going to talk about how neurogenetics can help our patients. I'm going to talk about Mendelian diseases. So that's where diseases run in families, transmitted from generation to generation, in the case of dominant disease, and ask about how that links in with sporadic disease when patients don't have a family history, particularly with two diseases that or areas I've been involved in. One is ALS, motor neuron disease, together with frontotemporal dementia, and how that links into patients with motor neuron disease that have no family history. The other condition is frontotemporal dementia, or PICS disease, a tau-related neurodegenerative condition, and how that relates to PSP, progressive supranuclear palsy, which is a sporadic tauopathy where patients have no family history of disease. And as I said, to illustrate that genetics is about people and places. So essentially there are only two sorts of studies that we do in neurogenetics. One is based on families, where we look for very, very rare variants that segregate with the disease. So our tools for this are to look at families, look at multiple members of the family with and without disease, look at the genomic segments that might go along with the disease, a little section of DNA that might be inherited in people suffering from disease, and then to hone in on the pathogenic change in the disease, the thing that actually causes the disease in these patients. And those are Mendelian studies. The other type of study we do is sporadic patients, and essentially this is very simple. We get a bunch of cases with disease, a bunch of controls, we look at a variant, and we look to see whether it's overrepresented in cases versus controls. And there are various methods of doing that. You can look at candidate genes, or now using whole genome approaches to look at variation across the whole genome. So here we're looking at variants that all of us possess. They're obviously not necessary, they don't always lead to disease, but there are risk factors which are more common in patients who suffer from these diseases. Okay, So this is a family which is an extremely important family, and this again really illustrates, particularly actually to the clinicians here, the clinical neurologists who are here, and to the students, how just studying in detail a very small number of people can give you insights into a disease across the whole of the world. So before I left Queen's Square, I wrote a grant to study this condition, which wasn't funded. And then when I moved to Wales, the first thing I did was to come across this family, in which many members of the family had been suffered from either motor neuron disease, ALS. In fact, do you use motor neuron disease or ALS? ALS. Yeah. So either ALS or this dementia, frontotemporal dementia, or Pick's disease. So many members of the family had suffered from this condition. And this has been a curse, if you like, on the family for many generations. Many generations of the family had suffered from this condition. And as is often the case, the research was really driven by one member of the family. So it was a lady who I met who was in her early 60s who had not been affected by this condition, but she'd seen 4 of her sisters die from it, she'd seen her mother die from it, and many nephews and nieces died from this condition. So this was something that affected her and her family very deeply, and she was driven to helping me to identify patients, to take blood samples, to have the appropriate consents, to try and find out what the cause of this condition was. So this was a patient we saw in this family. So this is a lady who was 46. She was a very house-proud lady. She looked after her house meticulously, paid all her bills on time. She began to have a deterioration in her social conduct. So she began to become disorganised, and she became untidy. She started being rather impulsive, so giving money away to people. It's one of the things that unfortunately we have a lot in the UK is people come knock the door and say we'd like to buy some double glazing, or we'd like to buy this insurance policy, and normally people will say no, go away, or words to that effect. But often patients with these conditions, say, oh yes, here, I'll have all the money, I'll sign up for this, and often end up spending a lot of money in the early phases of this disease. Her memory was relatively intact, so her memory for the day-to-day events was intact, but her social conduct was very much impaired. She was found missing to a nursing home at about the age of 47 because she really couldn't look after herself. She had no weakness, arms and legs were normal, speech was normal. She just had such severe deterioration in personality and behaviour. She had to miss a nursing home. I went to nursing home and visited her. She developed psychosis, so she had delusional hallucinatory state as part of the condition. She then developed about 3 years after the onset of the condition very brisk reflexes, wasting, slurred speech, and features of ALS. So the clinical diagnosis of this lady was a frontotemporal dementia, behavioural variant frontotemporal dementia, together with ALS, and she died about 4 years after the condition started in an akinetic mute state, unable to move 4 years after this condition had begun. So as mentioned, she was part of this very important family that went kindred. So this slide was made actually before she died. We identified 144 family members within the family. The mean age of onset was 42 years. The mean disease duration to death was 3.5 years. Some patients had frontotemporal dementia, some had ALS, some had a mixture of these conditions. At the same time as we were working on this, other groups around the world were working on other families and seemed to have this pattern of problems. So this down here is one kindred. 70% had mixed disease, about 30% had ALS, age onset 42 years. Very similar to other families that have been observed around the world, including one that was studied by my colleague Adam Boxer in San Francisco, who had a very similar pattern of motor neuron disease, ALS, and frontotemporal dementia. So we obtained blood samples from this family. We looked at genomic variation across the genome to ask which the raft DNA that was inherited by patients who had this condition was, and they shared a segment on chromosome 9p21. So all affected members of this family had a segment on chromosome 9p21 that essentially contained 3 candidate genes: IFT74, Sinanor 72 and this MOL2B, so 3 candidate genes in the region. We obtained autopsy material on this family, which showed TDP43 ecology. So TDP43 is normally a nuclear protein that's mislocalized in the cytoplasm in patients with ALS. And it's an RNA, thought to be an RNA processing protein which is mislocalized to the cytoplasm in patients with that condition. I should say that we had exhaustively sequenced all of the genes in this region to look at the DNA sequence in an attempt to find a causative change that caused this condition, and we failed to do it. So we were stuck. We were stumped on this family. We joined a consortium with NIH. We couldn't find out what the cause of the disease was for this family. At the same time, the second type of genetic study is being carried out, just looking at sporadic patients with no family history disease, comparing cases and controls. So this is work that was done by Brian Traynor. This is looking at patients from Finland with motor neuron disease ALS, and each dot within this on this picture represents a single variant in the genome, in which we simply looked at the frequency in cases versus the frequency in controls. And very simply done a chi-squared test to look at the p-value and the difference between cases and controls within these families on a genome-wide basis. These genome chips contain about 500,000 markers. The threshold for genome-wide significance is 10 to the minus 7. So within these sporadic patients in Finland with no family history of disease, they had an association between genetic variants in 9p21 as compared to, which was more common in cases compared to controls. So in sporadic patients, exactly the same region of the genome was associated with a risk factor for disease in this study as was segregated within the family. And this shows the genotype data. Essentially, for these variants on chromosome 9, the frequency in cases was about 30%. So 30% of cases had it, as compared to about 15% of controls. So obviously it didn't cause the disease in these patients, but was a risk factor for disease. So this illustrates the two different approaches, the case control approach and the family-based linkage and segregation approach. And actually both in this case are honed in on the same region. So I'll come back to this family and what happened next in a minute. So how do neurogenetics help our patients? The manifesto for what we do is very simple. Essentially, we find families with disease. We use those families to find a gene, and more exactly, the gene variant, the mutation that causes the disease. We use that gene variant to develop both cell and animal models for the disease. We develop new therapies based on that. We take those therapies back to the patient. Along the way, there are incredible side effects that come from doing this type of research. One is understanding the disease better. It was very debated whether frontotemporal dementia and ALS are the same condition or different conditions. We knew in this family that some people got ALS, some people got frontotemporal dementia, so there are two sides of the same coin. We knew just from studying this one family that these were two variations of the same condition. And actually we now know that ALS very commonly has frontal behavioural changes as part of the disease. We're able to carry out definitive diagnosis, genetic counselling, sometimes predictive testing to see whether people are going to develop disease later in life. The big question is whether this type of discovery can actually help patients with sporadic disease. So understanding whether these autosomal dominant families are also a good model for sporadic disease is absolutely crucial. Because literally hundreds of millions of dollars have started at this stage in developing animal models and cell models for disease based on genetics. So in Parkinson's disease, particularly synuclein and LRRK2, based very much on the families who identify these mutations, but do we really know that those are helpful and useful in patients with sporadic disease? So further work was done on this locus, this area which carries gene change. This is work led by my friend and colleague, Professor John Harney. So this does not project so well, but essentially in the Finnish study there was this raft of DNA that was shared by cases who had motor neuron disease, ALS, in Finland. And even across here in the variants across this region that were shared by these families. fact, the Gwen Kindred carried the same wealth of DNA as these patients over in Finland who had sporadic motor neuron disease ALS, and actually all of the other families also carried the same segment of DNA. So it looked like the same segment of DNA was both the risk factor for the disease and the cause of the disease. So we engaged in collaboration. We did a lot of next generation sequencing, so sequencing all of the variants, both the intronic, non-coding regions and coding regions, and eventually the cause of this was found. And I would say as well, when I started working on this, I struggled to get funding to do this. People said, oh, what's the interest of this family in the Welsh Valleys that has ALS? Why is this going to be important? It turns out the discovery made based on this family is the commonest cause of ALS all over the Northern Hemisphere. It's the commonest cause of ALS we currently know about. And the discovery of this made the front cover of Neuron. This is the hexanucleotide repeat expansion that causes ALS. So it turned out the reason that we struggled to find the cause of this disease is that we've been focusing on exons, coding sequences, and also been focusing on next generation sequencing, which produces short reads of DNA, 90 base pairs. And in fact, this is a massive expansion, which is quite difficult to detect with this technique. The expansion is in non-coding DNA sequence, and it's either in the promoter region or the first intron of this gene, which is still called C9orf72. It's a massive hexanucleotide repeat expansion. So normally, if we were to take blood samples from people in this room, measure the number of repeats in this area, we'd find less than 20 repeats. 20 hexanucleotide repeats containing 6 base pairs, GG, in people in this room. If you're one of the people in this family, then you'll have an expansion, usually of more than 2,000 repeats in that region. So it's a massive DNA expansion in that region which leads to disease. And this is illustrated with a repeat prime PCR. This is normal. This is a patient with disease where when you use a probe to the actual repeat region, you can develop you get this so-called hedgehog pattern. And it's also shown with the Southern blot, which is the workhorse of genetics 20 years ago. If we started off doing Southern blotting, we would have found this probably a lot more quickly. This is a control. This is the first of the 23 KB band, which represents the repeat expansion. So a massive expansion, which as expected, segregates with the disease. So in the Gwen family, every person who had the disease also had this massive DNA expansion in the C9orf72 gene. And this is the group that worked on this discovery: Adrian Waite, Nigel William, Jim Neal, and Derek Blake, and this was funded by the Motor Neuron Disease Association. So that was a very satisfying discovery. The family who were involved were overwhelmed with this disease, the cause of this disease being identified in their family. But what really made us fall off our chairs was how common this variant is. So this is a paper published in Neurology. This looks at the frequency of the expansion in sporadic patients with no family history. And in fact, you're not going to read these numbers, but the frequency of the expansion in patients with ALS is about 5 or 6% in the UK. So 1 in 20 ALS patients carry this expansion. And in fact, this is a disease probably of Northern Europe. The founder probably is from Finland, and this disease across Northern Europe and America probably represents migration of this person, this founder, across Northern Europe. So here are the frequencies, and as you move further south in Europe, the frequency of expansion in cases gets less. And in fact, if you look at other parts of the world, the frequency of expansion is very low. So in Northern Europe and the United States, the frequency is about 5%. There's only ever been one patient identified in the Middle East who carries this expansion. There have been very few people in the Pacific Islands or in Asia who carry this expansion. This is a very important cause of ALS in Northern Europe, but it's not an important cause of ALS in the Middle East or in Africa, as far as we know. I think there are a few kind of conclusions that you can make about that. One of the conclusions I would immediately lead to is that there must be other genetic causes in the Middle East and Africa which are equally important for people who live here, but also will give us different insights into disease and how the disease happens. And this is an absolutely crucial message that I want to get across today. So, does dominant disease explain sporadic disease? In this case, the same mutation was present in patients with no family history and in patients with familial disease. How can this be? How can it be that someone comes in with no family history and they carry an autosomal dominant mutation? One reason for this is penetrance, so the disease may not affect some patients who are 50s or 60s, so patients the previous generation may not have lived long enough to get the disease. The other reason is misdiagnosis, and this is something that I've experienced as well. So a patient with ALS, they carry the expansion, any family history of ALS? No, so they have no family history written down. Going through things a bit deeper, what about your parents? Oh yeah, my mum had Alzheimer's disease when she was in her 60s, and she died after 2 years, and she behaved very strangely, and she did all sorts of bizarre things. So actually, I'd missed that diagnosis in the mother. The mother probably had frontotemporal dementia, and actually this was a familial disease, but I hadn't picked up on it. And actually a very, very common misdiagnosis as well is psychiatric disease. So patients often diagnosed with psychosis when they're in the early phase of this disease. So this is important that dominant disease appears to be sporadic. Probably the most important gene we currently know about, particularly in this part of the world, is the LRRK2 gene, the LRRK2 gene, which is the commonest cause of autosomal dominant Parkinson's disease. This causes late onset Parkinson's disease, so patients who develop Parkinson's disease from their 60s onwards. In fact, if you carry this gene mutation in a family, then your risk of developing disease at about age 70 is about 30%, and by age 80 it comes up to 50%. So you can see that it's quite common that previous generations will not have lived long enough to get the disease. So it look like it's sporadic, but actually it's a dominant disease that's running in the family. This is this G29S mutation is a very, very rare and important cause of Parkinson's disease. And as I say, this is very common in this part of the world. So in fact, in the UK, it's 1% of sporadic patients, 2% of familial. In studies particularly done in Tunisia, I think, in North African Arabs and Algeria, 40% of sporadic Parkinson's patients carry this mutation, and about 30% of familial. So it's really a very common cause of disease. And this is important both in terms of potentially counselling your patients. So if you have a Parkinson's patient, there may well be the next generation of risk of Parkinson's disease as people live longer and get older. This is also a very important area for translational research. So many drug companies are trying to develop therapies that are going to be directed towards the G29S mutation. So actually they need lots of patients with this condition, and obviously countries like Egypt, which should have a large number of patients with this mutation, are also going to be an excellent place for drug companies to build links and start to do this type of research, trying to help patients with this mutation and to stop the disease from developing. Okay, second family. So this is another patient, kind of a similar story actually. So this is a lady in the early 50s who was referred to me with Parkinson's disease, not responding very well to L-DOPA therapy. So she developed an asymmetric tremor, stiffness, slowness of movement, bradykinesia. So she'd been prescribed L-DOPA, so dopamine replacement therapy. She had done extremely badly, hadn't cut with a movement disorder, and her behaviour had changed markedly. So she began to swear inappropriately at people. She became rather disinhibited. She'd go up to people and make sexual remarks or make remarks about their weight, which she normally wouldn't have done. So again, this is a patient developing social disinhibition as part of a frontal lobe syndrome. She also developed compulsive eating of sweet foods, which is very common in this condition. So this lady was part of this family. In fact, her mother had been diagnosed with schizophrenia. So again, occult family history. The family history wasn't obvious unless you scraped the surface and went deeper into the family history of what had gone on in the family. She was institutionalised in psych hospital and died. She had no effective siblings when I first started working with Gwent, but then two further people in the family became affected, and there's some very distressing things that happened within these families. So in particular, this gentleman who developed this disease was at his sister's funeral, and at the sister's funeral it became apparent that he was behaving inappropriately, started laughing, dancing, singing at a time when everyone was very sad and sombre about their relative's death. So it became apparent at the funeral that he was behaving inappropriately, and in the early stages of the same disease that had just killed his sister. tragic situation. So this family has frontotemporal dementia linked to chromosome 17. The gene for this is the tau gene. This was cloned in 1998 by a group led by Mike Cutton. Briefly, the tau gene is alternatively spliced, so it has some protein coding sequences, exons that can either be included or not included in the RNA transcript. In particular, exon 10 is alternatively spliced, and there are different isoforms of the gene that are converted to RNA and then to protein, which means there are 6 different isoforms in the human brain, which are varied by the inclusion of exon 10 or exons 2 and 3. These variants, these are perfectly repeated sequences that bind to microtubules, so perform a crucial function in stabilizing axons and microtubule transport. These are known as repeat domains. So these are 3 repeat forms of tau. These are 4 repeat forms of tau. It turned out that the mutation in the family that I showed in South Wales was one of the mutations that had been described initially in the FTD17 paper. In fact, not a mutation in the protein coding sequence. There's a mutation of the +16 part of the intron. So what this does is the intron exon boundary normally forms a stem loop that sticks together, and this serves to stop exon 10 being put into the final transcript of tau. This mutation opens up that stem loop and it drives the production of tau that contains exon 10, the so-called 4 repeat tau. You can see in the different families, this is RNA analysis taken from the brain. This is normal controls. You have a 1-to-1 ratio of 4 repeat, 3 repeat tau. The family have this splice site mutation. There is an excess of 4 repeat tau, so there is a shift in the ratio. So this is normal sequence tau, but a shift in this ratio can cause the disease. As it happens, this mutation, this +6 mutation, is a Welsh mutation. So the founder comes from North Wales, and families with this mutation have been identified in Australia, North America, Wales, London, Manchester, all over the world, but they're all related to a common founder from North Wales. So this is a Welsh disease which is spread out around the world. The pathology of these diseases involves tau deposition, both in neuronal glial depositions in the brain. These patients have very marked frontotemporal atrophy. So on MR, there's atrophy of the temporal lobe, and there's a dilation of ventricles in the frontal lobe. So this is an autosomal dominant Mendelian tauopathy due to rare mutations. There is another condition called PSP, progressive supranuclear palsy, which is a sporadic tauopathy. People don't have a family history, but have very similar pathology post-mortem with neuronal glial tau deposition. The tau deposition is particularly in the brainstem and in the basal ganglia, subthalamic nucleus, and goes pallidus, and this causes a very typical clinical syndrome of gait problem, gait and balance disorder. In fact, this year is the 50th anniversary of the original description of PSP in archives of neurology by Steele, Richard and Shusky. And the pathology is very similar to the pathology of FTD17. So in fact, it turns out, again, that common variants of the tau gene predispose patients to developing PSP. So a very similar story to the ALS chromosome 9 association. So this is the frequency in controls of the H1 haplotype, which is about 80%, and in cases it's about 94%. So obviously this haplotype, these variants in the tau gene don't cause the disease, because a lot of controls have them, but there must be a risk factor for the disease, because they're more common in patients who have the disease. It turns out that this association is related to an inversion in the tau lesion. So the H2 haplotype, which is an inverted DNA sequence as compared to the H1 haplotype. Interestingly, the frequency of the H2 haplotype is very different in different parts of the world. So this is related to when I was working on this originally, and this is using a proxy for the haplotype. And actually in the UK population of PSP patients, 90% of them had the variant, 73% of controls, a highly significant p-value association. In Japan, in fact 100% of cases had it, but so do 98% of controls. So it's not possible to detect an association between the variant of the disease in the Japanese population because of the background frequency of this variant allele. So some people actually, when that was reported, described this as tau not being important in Japan. I would disagree with that. I would say that tau is probably equally important for PSP in Japan, but we just can't detect it because of the background genetic structure of the population. So different populations allow us to detect different things. And there may well be things that we have missed in studies in Western Europe that we can detect in Japan or in Africa or in Asia, and we haven't yet studied those patients to uncover those associations. And this actually shows, again from my colleague John Hardy, this shows the variation of the H1H2 haplotype around the world, looking at the frequencies in control populations around the world. In Africa, in Southern Africa, there are virtually no patients with the H2 haplotype. It becomes more common in Northern Africa, and most common, in fact, in Icelandic populations and very Northern European populations. So the question is, how can this cause the disease? Are the same mechanisms that are responsible in families also responsible in PSP? So I mentioned that RNA splicing is important. To try to uncover this, we started to look at RNA from the brains of patients with PSP, anti-normal controls, look at the effect of variants. The principle of this is very simple. This is allele-specific expression. We take a variant which is different. In a patient who's a heterozygous, they have one H1 allele and one H2 allele, and it has to be in the RNA, so you have to be able to measure it in the RNA in the brain. And at a genomic level, you look at the DNA, there's a 1-to-1 ratio. If there's a difference in expression, then you see an imbalance in the RNA. So you get the RNA, you convert it to cDNA, compare the RNA cDNA to DNA from the genome, and if there's an imbalance, that shows that one allele is being transcribed differently. So there are effects which are changing the transcription of that allele. It turns out there is no difference between the amount of the total tau that comes from the H1H2 haplotypes, but there is a difference in the amount of full repeat tau. So that in patients in the PSP risk haplotype, there's about a 10% increase in the amount of full repeat tau. So there is a subtle change in the alternative splicing of the tau gene related to this common variant. So we come to an idea that there are sort of different pathways to disease. You have pathways to disease which are sporadic, so tau splicing, a very subtle change in splicing. There are other risk factors now identified, leading in some way to tau aggregation, and there are various downstream effects which lead to neurodegeneration. We also have these very rare mutations in these families that lead to differences in tau splicing, to a much bigger effect, much earlier onset, but essentially the downstream effects are the same. So we have some synchronization between disease mechanisms in sporadic and early on disease. So a subtle change in PSP, major change in the mutation. So linking this together, this again is this is from a review by Andy Singleton, talking about the way that bits of the genome can predispose to disease. And in a nutshell, you can have rare changes which make a very big difference in the expression of the gene, which cause disease. You can have coding mutations, which cause a large amount of risk. You can also have very subtly change expression to a very small effect. And recapitulate the same mechanism, but to a much lesser extent in sporadic patients. So a spectrum of risk is sporadic with early patients. This is the first part of my talk. Doing pretty well, 33 minutes. So I've covered the general principles of genetics. Basically, very simple. There are two things we can do. One is look at families that are inheriting disease and look to see a segment of the disease. The other thing is to do association studies where we just look at cases in a box, controls in a box, and look at differences between them. How can neurogenetics help our patients? We hope this is going to help us understand the mechanisms that lead to the disease and develop new treatments. How does Mendelian disease relate to sporadic disease? You can either have the same mutation in sporadic patients and familial patients, which happens because of reduced penetrance, most likely, or you can have the same mechanism with subtle differences with sporadic and familial patients. And lastly, as I said, genetic research is about people and places. Founder effects are very important. Some diseases are more common in Finland or Wales, which which someone has passed on the genes to other people. Background frequencies are very important when you do case control studies. We will discover different things in different parts of the world. So, that's the first talk. I'm going to drink some of my coffee and take any questions. Go ahead.
Thank you, Dr. for your talk. I was just wondering about saying that this disease started in genes and this is the origin and then it's so how do you know that this is the origin?
Yes. So the way that tends to be done is that so that when a mutation arises, it arises on a haplotype. So there's a background raft of DNA that it arises on. You can look at that by looking at all the gene types along that. As that gene mutant is passed down, you have recombination events which serve then to make the haplotype around it shorter and shorter. So essentially, where the part of the world where an individual has the longest haplotype, and then you can do a sort of a family tree of all the haplotypes. You see it getting shorter and shorter because of recombination events that serve to make the haplotype shorter. So so that's genetic evidence, but obviously sometimes there's historic evidence. So we know that actually from Wales, a lot of people emigrated to Australia. Actually, they were sent to Australia for being criminals. A lot of people emigrated to America. So we know historically that these links exist. And actually, yeah. So, but it's the genetic markers around and how they're shared, how long the segment is. It's the main part of the piece of evidence. Okay. Thank you.
I want to ask you, the epigenetic changes will be epigenetic changes will be gene design responsible for some genetic changes if other in the occurrence of sporadic cases?
That's I think that's a very interesting question. I think that epigenetics is quite difficult to study. I think that obviously a lot of what I've spoken about is about gene expression and about ways that gene expression is controlled. And clearly methylation of motor regions and epigenetic change is absolutely crucial for regulating gene expression. And as you know, epigenetic things can be inherited, or they can be things that relate to environmental changes that change gene expression. So I think that the evidence of this at the moment is limited, but I think that there will be epigenetic changes that can be identified. And you see that, a lot of people are doing sort of genome-wide studies of methylation and epigenetic changes. For me, I'm a very simple-minded clinician who does some research. So for me, the simplest thing is to look at families and to ask the question, are there epigenetic changes that recapitulate what happens in the families? Because the families are actually rock solid. We know for definite that's what causes the disease. It's completely beyond dispute and argument. So actually I think trying to link it together with, for example, the tau locus, where we know lots of mutations, or the synuclein locus, where we know there's triplication that causes the disease. You have 4 copies of synuclein gene, which causes Parkinson's disease. So my bias would be to focus on those regions. So my colleague, the other thing that I think is important about epigenetics is that we know that epigenetics is different in different organs of the body and different parts of the brain. So actually my colleague Nigel Williams, who's grey-haired gentleman one of the grey-haired gentlemen in that picture, is now doing some work looking at epigenetic methylation changes in substantia nigra as opposed to different brain regions, to try and understand more about the regional variation in the epigenetic changes. So I think it will be. I think it's hard to study. Difficult to study. Okay, any other questions?
Thank you very much. There is a link between particular dementia and diabetes. And this opens a big question whether you know the explication one pathology, different type, and so different physical presentations. In dealing with gene translation, gene expression, and gene clearing, which is kind of the similar process, opens the gene here, if failure different continuum with different cases and with different situations and clinical presentation, different ones. خصائص الخلية يعني affecting the affecting frontal lobe, temporal lobe, frontal dementia, affecting the motor system, ALS, and so on. What's the
Yeah. So I think I mean it's one of the biggest questions is why disease is affected from visceral brain. Obviously, these families, we know that some people get ALS, some will get FTD. They've got the same mutation. So why is it that one person gets frontal dementia predominantly and one person gets ALS? You know, why is one person frontal lobe affected, the other person the anterior corn cells affected? And I think we're trying to look at that with a genetic approach. So actually do a case control study of mutation carriers, look at differences in FTD and ALS predominance. So see if we can find any genetic variants that change the way the disease presents. But I'm sure there will be other things to do with epigenetic mechanisms, expression mechanisms, environmental factors that will determine the variability in disease, which is very, as you mentioned. So I think this is going to be a very active area of research, but again, very difficult to study. I think actually I think one of the most important things to study a lot of these things is post-autopsy material, and actually being able to look at autopsies and extract RNA and DNA from different brain areas. I think is going to be very important in helping to understand these things. Okay.
With regarding gene mutation, you said that there is some facility to block gene mutation.
One of the things with this RNA, so I didn't really talk about how the RNA expression causes disease. There are a few different theories about that. One of the main theory at the moment is that this massive RNA expansion actually sucks in RNA splicing factors and causes misregulation of the whole family of genes based on the RNA expansion sequestering splicing factors, which is exactly the same mechanism as in myototic dystrophy. So myototic dystrophy is the same thing happens with suppression splicing factors. So many people are trying to identify anticellular nucleotides that can turn off the expression of the expansion and to stop the expansion from causing disease. So I think that's a very active area of research. Okay, second talk. This is now particularly focusing on Parkinson's disease. This is a research program I set up when I went to Cardiff and still carrying on. This really is based on a pretty simple question, which is we know Parkinson's disease is an age-related condition. In the UK, the average age of onset is 68 for Parkinson's disease. So a very simple question: if someone walks into your clinic, age 25, and has developed Parkinson's disease, that is a very, very unusual thing to happen. So just at a basic level, there must be a good reason for that. That person must have some sort of exceptional environmental exposure or some sort of exceptional genetic factor that causes the disease. So the idea is that if we study these extreme outlying cases, we can learn more about the disease as a whole. So early onset Parkinson's disease, how does it happen? So our approach to this was really to a sort of clinical and then genetic study. So I'm going to talk a bit about a community-based study of Parkinson's disease we did, a little bit about a family study, some molecular stuff, ongoing work, and how I think this may become relevant to therapy, and how we may link different things together. So in South Wales, the best way to do any epidemiological study is try to recruit all of the patients in the community who have a disease. So what we did in Cardiff is we went to every general practitioner or family practitioner in Cardiff and asked for permission to look at their records for people who prescribed Sindec-Madapar with diagnosis of Parkinson's disease, Parkinson's syndromes, to try and capture every patient in the community to have a community-based survey. We know in the UK that we sit in specialist clinics, you sit in Queen Square and see Parkinson's disease, you have a very, very different idea of what Parkinson's disease is as compared to what it is out in the community. Because you see very highly selected patients that are coming up to that point. So to understand the disease properly, the best thing to do is a community-based study. If you talk to an epidemiologist, they'll say what you should do is go and knock on doors, house to house, and see everything, this is too much, do that. But that's what they like to do, is their gold standard study. So we looked at every patient with Parkinson's disease in Ireland. We recruited, actually through this study, we recruited about 600 patients all told, with DNA and clinical data. And to cut a long story short, what this guy here has got, Michael J. Fox, developed Parkinson's disease age 29. What he's got is very, very unusual. So in our survey, we only found 1% of Parkinson's patients who developed their disease below the age of 40, 3.5% below the age of 45, 5.5% below the age of 50, 31% below the age of 65. So this early onset disease is very unusual. The clinical features we looked at, we wanted to know whether early onset disease was different to late onset or the same. In fact, many clinical features of Parkinson's disease have a gradual change between early onset and late onset patients. So, for example, tremor-dominant disease, patients who present primarily with tremor, is much more common in older patients, whereas patients without tremor will only have bradykinesia, common in younger patients. This ratio of presentations is something that progressively changes as you move through different age bands. So, elbow-induced dyskinesia is involuntary movement in response to elbow treatment, much more common in young patients than old patients. So, about 40% of early onset patients have this sensitivity of the striatum to L-DOPA, these plastic changes in the striatum that lead to this dyskinetic response, whereas this is much less common in patients with late onset disease. One thing that we did find different, it seems to be substantially different between early onset and late onset disease, was dystonia in Parkinson's disease, so fixed muscle, abnormal muscle contractions that occur in Parkinson's patients. Often first thing in the morning, Parkinson's patients get dystonic foot posturing or as their medication wears off. These phenomena were much more common in early onset patients than late onset patients, which is interesting. We know that if you give schizophrenic patients dopamine blocking drugs, younger patients much more likely to get drug-induced dystonia, older patients much more likely to get drug-induced Parkinson's. So there's a difference in the age and the dopamine system between early and late onset patients. So this is a family study. Again, illustrating why family things are important. This is published as movement disorders, but the video is not published as movement disorders. This lady. So this is a boy, symptom study age of 8, who dragged the left leg, then spread to the right leg. Initially thought to have a medullary tumour, and then in his teenage years was known to have lack of facial expression, rigidity, slowness of movement, and the diagnosis was revised to an extrapyramidal disorder. He, in his early 30s, he saw the great Professor David Marsden at Queen Square, who made a diagnosis of a pallidal atrophy syndrome. So he didn't think this was Parkinson's disease, he thought this was a degeneration of the globus pallidus that was causing this dystonia Parkinsonism syndrome. So I'm just going to, so we were aware of this patient. What are we going on with him? Very early onset Parkinson's disease. This is the youngest patient we identified, age of onset, age 8. We did actually meet the criteria for Parkinson's disease. Then we saw his sister, and his sister didn't like to go to the doctor. The reason being, when she was in her late teenage years, she developed some symptoms, and she was told by a family practitioner that she was just copying her brother. And it was all psychologically based. They said, there's nothing wrong with you, you're just copying your brother. And she was sent away. And she actually never really went to doctors after that. But somehow she did obtain a prescription of triacetylphenidol, an anticholinergic drug, which she was taking to treat her symptoms. So I'm going to show a video now. Well, actually, is there a sound with the speakers? Is there a sound cable? I'll check. I don't think if there's a sound cable, probably not. So she's going to talk about the symptoms anyway. I'll tell you what she's saying. So this lady's given a consent to the video for teaching, but not for distribution. So can you turn off the video? Is the video thing playing? Can you turn off the video before I just show explaining. Just for one slide. Is a variant call on the genotyping chip. So you can either be homozygous, you've got one allele from your mum, one from your dad. You can either be homozygous for the common, heterozygous, or homozygous for the rare. So the call you can have is either AA, AV, or VB. The terminology is used for this. We also have on the bottom the B allele frequency, which is a measure of the intensity signal that's coming from the chip as the laser scans across the chip. In this region of chromosome 6, there is a drop-off of signal for these patients, so they lose the signal of chromosome 6, and you don't see this here, but the automatic variant caller doesn't know what to do. So there's a sort of splurge of genotype calls that don't really cluster around the AA, AD, or BB region. So the machine is foxed. It doesn't know what to do. The reason for this is the patient has two deletions. So it has two deletions of the Parkin gene, one from mum, one from dad. The deletion of one is exon 2, deletion of the other is exon 2 and 3. So the patient, these brother and sister, have a massive loss of function. Actually, you can turn the video back on now. I don't know if you do that, but I've seen the video. You can turn it back on the video.
So what?
You can put it back on, it's fine. So these patients have a complete loss of function of the Parkin gene because they've got a knockout of both mum and dad's copy of the Parkin gene. So they've a massive loss of function of this protein. This is originally described in Japanese families. This is paper from Nature, 1996, when consanguineous families in Japan were used to map this disease and to map the Parkin gene. Some people think that this is different from Parkinson's disease. This is a review paper from Eric Alsmog pointing out this patient has a much more benign course, much less likely to cause non-motor symptoms, much less likely to cause dementia, gait and balance problems, but still causing a nine-wall disease of touch nigra, elevated responsiveness. In fact, I'm not sure whether this will culturally, does anyone know who that is? Without reading the top. So this is the lead singer of Black Sabbath, a heavy metal group in Britain, called Ozzy Osbourne. And there's a movie, there's a TV show called At Home with the Osbournes, where it's an American TV show where he's with his wife at home, and it's kind of funny because it's kind of domestic. He used to be in this heavy metal band, and he used to bite the heads of animals on stage. That was one of the parts of his act. And then he's in this domestic show with his wife. And anyone who's seen the show will know that he shuffles around. He's got a very stiff posture. He's very shaky. And I had assumed that the reason that Ozzy Osbourne had Parkinsonism was that he'd been consuming a lot of drugs in his younger days, and that's some sort of drug abuse Parkinsonism. But actually, he also has this Parkin disease. So this is from the web. It says Ozzy Osbourne fights Parkinson's disease, brackets Parkin syndrome, with his music. So this is, I think if you look at the table thing on that TV show, this is not like typical Parkinson's disease. This looks like a different syndrome. We know in Parkinson's disease that there are mitochondrial abnormalities. So this is work from my current colleague at the Royal Free Hospital, Tony Shapiro, from the early 90s, looking at platelets, mitochondrial function, and showing there's a specific deficit in platelet complex 1 in patients with Parkinson's disease as compared to controls. I think one of the key things about this figure is to observe that there is a very big overlap between controls and cases. So although on average Parkinson's complex 1 is lower than controls, in fact some have in the normal range, some controls are quite low in complex 1 as well. So this is not absolute, this is an overlap. It turns out that Parkin is very important for mitochondrial function. So in fact, when mitochondria get damaged, PIC1 is normally held and released from the mitochondrial membrane. If you damage mitochondria experimentally, PIC1 is stabilised in the mitochondrial membrane. That recruits Parkin, which then triggers a series of downstream effects which lead to mitophagy and clearance of the abnormal mitochondria. So Parkin is crucially important for mitochondrial quality control. And in these skin biopsies in patients with loss of function, they have a specific loss of function of complex 1. So I think this is a fascinating disease model. And I think this obviously, I know some of you are working on toxin models, and And this really is kind of illustrating that we can have both genetic models and toxic models that work on the same types of systems. In this case, mitochondrial complex 1, mitochondrial quality control. So you can have Parkinson's disease or a form of Parkinson's disease either because of toxic damage or because of genetic factors. And it's a similar story that you can have ion channels. With ion channels, you can either have mutations in the ion channel or you can have antibodies that block up the ion channel. I think one of the reasons that this is really important, this is from a paper in Neurology looking at disease-modifying therapies in Parkinson's disease. This is looking at codon Q10 in Parkinson's patients. There is no treatment effect, so potential mitochondrial therapy in Parkinson's, there is no treatment effect. For me, the failure of these studies is to lump, well, there are two failures. One is to lump all Parkinson's disease patients together, like they have the same mechanism, when they may have very heterogeneous mechanisms. So it may be that a subset of patients respond to complex to codon Q10 and the others don't. So I think lumping all patients together is a mistake. We need to dissect out the disease mechanism. The other thing is that many of these studies don't have any tests that tell us whether we've improved the system. So they don't have CSF or MR spectroscopy or skin biopsies. If you're trying to improve the system, this doesn't tell us that mitochondrial therapies don't work, because we don't even know if this has improved mitochondrial function. So we need to know that we've engaged the target and improved the system. We also need to know we've got the right patients. And I think understanding disease mechanism is obviously crucial for this. So a little bit about genetics. So we looked at all these genes in the Welsh population. We did a systematic mood disorders, systematic review all around the world of different disease genes. About 10% of Parkinson's early onset patients have Parkin mutations, about 4% of PIP1, PIP1 very rare. All of these, well, Parkin and PIP1 definitely are very important for mitochondrial function, mitophagy, and are on the same pathway. In fact, in Asian populations, PIP1 is more common, so there's a found effect in the Philippines, the L347P mutation, which is very common in Filipino populations, and is a very common cause of early onset Parkinson's in that population. So again, different diseases, different mechanisms, different parts of the world which we can learn. Our aim is to find new genes for early onset Parkinson's disease. This is homozygosity mapping, homozygosity analysis. This looks again at mum and dad's genome that all of us carry, and looks for segments where mum and dad's genotypes or the alleles we've got from mum and dad are the same as the homozygous. So this is a marker of parental consanguinity. So if your parents are first cousins, it's more likely you have one of these homozygous stretches. And we know that homozygous stretches are a risk factor for recessive disease, because if there happens to be a rare variant that your mum and dad have, and they are first cousins, then it comes together and it causes recessive disease. So we showed in the UK population that there's excess of homozygosity. So across the genome, early onset Parkinson's patients are more homozygous. Their parents are more closely related than patients with late onset disease. We try to use that to find new genes for Parkinson's disease. So we looked at a series of patients with homozygosity. We've got these shared segments of the genome. We've used whole exome sequencing to sequence all of the exome, all the protein coding bits, looking for rare variants that we could deduce might cause Parkinson's disease. And we have a replication series as well. To cut a long story short, that's been unsuccessful so far. And the main reason for that is that each person carries so many variants that it's very, very difficult to see the link between or accurately. So each exome will contain 5,000 deleterious variants, 500 be rare. You have 1,000 PD genomes, which we do have. So we have 500,000 rare variants. Each genome representing 25 times by chance. You can see the statistical constraints of finding something which may be very rare within this population is very, very difficult. I think our approach to this is that we need to have younger cases. We need to recruit more patients with families who are consanguineous, with their parents are related, because these are so valuable for learning what's going on with the disease mechanism. We need to recruit more family members. We need to have actually one unaffected brother and sister. This makes it very much easier to do exome work, because you can subtract one from the other and work out what's pathogenic, what pathogenic change is. I suspect that some of these slightly older patients we recruited, who are in age 40 onwards, they may have quite complex disease. We have to notice that now. They're making polygenic, they're making multiple risk factors come together to cause the disease. So, in summary, I briefly outlined the fact that we've done a community-based study of Parkinson's disease. We think about 1% in the UK, we think about 1% of Parkinson's patients have disease before the age of 40. I've shown a family study with Parkinson's disease, showing that family effectively has a knockout of Parkin. So many of you will know about knockout models in flies or worms. This is a human knockout of the Parkin gene. This is the phenotype they get. I've explained how this links to mitochondrial function. I've explained that about 10% of early onset cases are currently explained. We have ongoing work finding new genes. We need to recruit more early onset familial contact with cases. And ultimately, I think the direction of travel here is going to be helping us design new therapies for our patients, which I think, for many of these things, if I can sort of try to draw both my talks together, an overarching thing, we have a blending of mechanisms from sporadic disease, environmental factors, probably epigenetics to come, Mendelian factors. These things are all going to blend together to give us ideas of disease mechanisms, but also understanding that epigenetic disease, that Alzheimer's disease and Parkinson's disease are probably not one disease. They're probably different diseases within them that will have different mechanisms. And hopefully we can use this to develop new therapies and treatments for our patients. So thank all of you who worked with me on Parkinson's disease project, particularly Mary Ratchie, who did the community-based work, deep query, we ran our database, and then went on to do a PhD actually on paraquat exposure in Sri Lanka, and his role in Parkinson's disease. Stephen is South African postdoc, who's doing analysis. Yogesh Shlomo, Jewish epidemiologist doing analysis. So I think we have quite a cross-cultural group, even though we're based in the UK. Funded by many different sources, particularly Medical Research Council, Parkinson's UK. So that's it. Thanks very much. So I think I've got time for maybe one or two questions. Sorry. Function. Yeah, the easiest way is to take a skin biopsy from the patient and then to grow the fibroblasts in the lab. And actually you can do mitochondrial function of fibroblasts in the lab very reproducibly. You can show the Parkin and pigment on patients. You can do very beautiful studies. Actually, don't measure complex 1, just measuring mitochondrial membrane potential, looking at mitochondrial morphology. You can do beautiful studies on fibroblasts. The problem we've got now in the UK is if you say you're doing these studies on fibroblasts, which are easy, patients are happy to donate samples as well. And they're, in fact, so many of our samples actually, we have donated to the Coriell repository. So these are available to people as an open access resource. So if someone here or another part of the world says, I'd like to study a Parkin patient, you can write to Coriell, and they will send you something from our patients, which we have made available to people all over the world to work on. The problem we've got with our grant funding bodies now in the UK is when you say you're doing this type of work, they say, oh, well, this is useless. You should be doing induced pluripotent stem cells to make your own models. Unfortunately, that takes about 2 years and an army of postdocs and students to reprogram these cells and to generate new own models. So this is very labour intensive, and we've written back to reviews saying we don't know what you're talking about. The cost of doing this is enormous. But there's some nice work on actual cyanogen triplications in neuronal cells that have been derived from fibroblasts that have been reprogrammed. So this is probably, I think the fibroblasts are the best thing at the moment. We don't have a good biomarker in blood. We don't have a good biomarker in CSF. MR spectroscopy, a bit uncertain. We'd like to explore that. Also, that'd be great to do a scan and look at ATP levels and reliably say what the state of mitochondrial function was in patients. But at the moment, I'm not sure that's quite there yet. So. Okay. Yeah. Yeah. I think it's very interesting. I think, well, the story here is that obviously encephalitis lethargica basically died out certainly in the UK in the 1930s and 40s. There were still cases being identified. So my colleague Andrew Lees, my mentor, wrote up a series of cases, and we had some patients that were admitted with fever, psychosis, often a orofacial dyskinesia, and then developed Parkinsonism and other movement disorders. And I showed the videos to people, and Andrew Lees and Gavin Giovanni said, "Oh yeah, we think this is probably encephalitis lethargica." But actually, then, so that was about 8 years ago. But then about 5 years ago, or 4 years ago, we had a researcher from Oxford come give us a talk about the NMDA receptor. And I looked at his talk and the video, and I said, yeah, that's what the patient has. And actually, that patient had the NMDA receptor antibody. And actually, many of the cases of encephalitis lethargica now seem to relate to this NMDA receptor. So I think antibodies are very, very important. And probably there are many other antibodies to be identified which are important in causing these syndromes. We know that the NMDA receptor antibody is particularly linked to teratoma. Again, we had another patient where it seems to be one of these molecular mimicry things with teratoma. The patient had a CT of their pelvis, said to be normal. I said to the radiologist to be sure that it's normal, and he said to me, "Oh, actually, now you come to mention it, the patient has teeth in their ovary, so she has a teratoma in the ovary, and that was taken out. That was the cause of the disease." So I think this is very important. There are some metabolic things that cause rapid onset. There's a gene for rapid onset Parkinson's dystonia, which is an ATPase, I think, that causes that disease. So, but I think we have to keep studying these patients. So, my advice, my advice is sort of to have the ethics approval, to store biosamples, to store DNA, to store serum, take videos, have consent, and these patients that you see will be a goldmine for making future discoveries and future research. Because undoubtedly you'll see things here that are different from what we see in London. We probably see things in London differently here. But these patients are a goldmine for discoveries, and I really think you're backing the samples. And I know you've done this with the New Genetic Network, you've started to do this, but I think you need to keep, you're working on this, you need to keep a tight hold on what's happening, and keep on asking the question about why did this patient get this disease, and we'll get more and more answers, I'm sure. Okay. Right, we'll switch to abstract courses.